Again, what we'll do today is we'll continue on from where we were yesterday. And what today's theme is going to be is ventilation and the structure and function of the thoracic cavity. Now, what we mean by ventilation is simply breathing in and breathing out. And as we work our way through this, one of the things we're going to find is that ventilation has two components to it, an active component and a passive component. And the ventilation, at least while sitting at rest, the ventilation active component is going to be generated by the contraction of the diaphragm. So in order to understand how that works, we have to spend a little bit of time drawing out the structure of the thoracic cavity and the association of the lung with the so-called pleura. And what we'll do is we'll basically draw the pleural sac and show the pleural sac surrounds the lung, much like the pericardial sac surrounded the heart. But here now, this pleural sac is going to play a really important role in ventilation because it's always going to be inside the pleural sac, inside the pleural space, there's always going to be a negative pressure there with respect to the outside. And as we move our way through this, we're going to see that it's pressure differentials which are going to allow for the movement of air in and out of the lung. The first story here is going to be the idea of the diaphragm. And the diaphragm is simply a skeletal muscle. Evolutionarily, it's related to the body wall muscle. The skeletal muscle is innervated by lower motor neurons. The cell bodies of these lower motor neurons are located in the cervical spinal cord. I think it's C2 to C5, and they're all found in the ventral horn. All to point out that this is common, garden variety, ordinary lower motor neurons innervating the skeletal muscle. And the interesting thing about this is we're going to see that when the lower motor neurons are activated, and when the lower motor neurons are activated and they cause contraction in the diaphragm, what that's going to do is it's going to increase the volume of the thoracic cavity. By increasing the volume of the thoracic cavity, we're going to see that we're going to generate a slight vacuum. Now, that vacuum can be formalized by Boyles Law. And Boyles Law is ancient. It came up in the 17th century. And it's an ideal gas law. And for our purposes here, what it says is P1V1 is equal to P2V2. And we're going to say that P1V1 is the ocean of atmosphere around us. So that's not going to change. That's going to be fixed. But P2V2 is going to be referring to the volume and pressure inside the lung. So you can imagine here, if you increase the volume, what's that going to do to the pressure? If the volume goes, to keep this thing equal, if the volume goes up, the pressure has to go down. So now we have a pressure differential. We have a pressure differential such that the pressure in the atmosphere is greater than in the lung. And once you see a delta P, you have a resistance and a flow. Yesterday we talked about the various resistances with the diameters or radii of the bronchioles and the bronchial tubes, and today we'll simply see how we generate that delta P. But as we've talked about so many times in here, what we do when we contract muscles, we shorten the sarcomere. But there needs to be a mechanism of restoring that sarcomere length to its original position. We talked about that in skeletal muscle, where we emphasized the importance of antagonistic muscles. We talked about that in the heart, where we said it was the low pressure return, the filling of the heart, which re-stretched those sarcomere lengths to their original position. We have the same problem here. If you put your phone away, I'd appreciate it. And we have the same problem here, where we're going to have to return that sarcomere length of the diaphragm back to its original length. And how are we going to do that? Well, it turns out that there's a passive recoil, a passive return to the original volume of the lung. That passive recoil has a number of different sources. One of them is elastin. The extracellular matrix around all of the alveoli will contain an extracellular series of proteins. And in a normal, healthy lung, much of that extracellular protein is elastin. So when you stretch it out, it's springy and comes back to its original shape. But there's another, even more profound source of this energy, and this is surface tension. And one of the ideas we'll try to think about here is how small the alveolar space, in fact, is. Each alveolus is in the neighborhood of about 200 micrometers across. These dimensions are small enough that the surfaces of the type 1 pneumocytes, the pneumocytes we talked about yesterday, can interact with one another. They can interact with one another through the surface tension of the fluid. So if there isn't some detergent in there, what will happen is the walls of the alveoli simply collapse and stick to one another. The way biologically we solve that problem is that we generate a detergent. The type 2 pneumocytes generate a kind of detergent, phosphatidylcholine. You remember phosphatidylcholine is one of these classic phospholipids with a charged head and an uncharged tail. And what we'll see here is that these phosphatidylcholine molecules will insinuate themselves in between adjacent water molecules, break up their surface tension, and then reduce the surface tension. But we'll also find that as we expand the alveolus, it gets larger and larger and larger during expansion when we breathe in, that these phosphatidylcholine molecules will get farther and farther apart. And as they get farther apart, their relative concentration decreases, and the water molecules now return to their more original situation of surface tension, their dipole moments interacting with one another, generating a force to snap these, the structure of these alveoli back to their original position. That will bring us to a kind of formalization of compliance and elastic recoil. These, if you look at this mathematically, these are just the inverse of one another. So the higher the compliance, the lower the elastic recoil. The higher the elastic recoil, the lower the compliance. And we'll talk a little bit about that. So let's start here with the structure of the lung in the thoracic cavity. We'll start with pretty much the description we had yesterday, where there's the larynx and the trachea branching into the bronchi, and then these bronchi further branching with their secondary and tertiary and quaternary branches. And so forth. And we can do the same for the other side. And then around that, in blue, I'm going to put the limits of the outer wall of the lung. I'll just put it in a dotted line. And we have two lungs, and so there's the left lung and the right lung. I'll make this a little smaller because I need to put some other structures in here. Now, you'll remember that when we talked about the way the heart fitted in the thoracic cavity, we said that there was a lining of mesothelial cells attached to the wall of the heart and another lining of mesothelial cells attached to the body wall. And those two then made up the pericardial sac. We're going to see the same arrangement here, where again, and I'll show these in red. Well, let me use purple. So that there will be a series of these purple cells right along the wall of the lung. These are in fact going to be mesothelial cells. The mesothelial cells are going to form a continuous layer all along the wall of the lung. And so I'll sort of extend these out quickly. And as we've seen before with the mesothelium, as with an epithelium, will be attached to the underlying tissue by a kind of basal lamina. And so the mesothelial cells are attached to the wall of the lung by a basal lamina that I'm showing here with these sort of red hashes. But the layer of mesothelial cells then folds back on itself. And then there'll be another layer of mesothelial cells whose basilar membrane is attached to the body wall. So then all of this red hashing in here is meant to be basal lamina that attaches the mesothelial cells to the body wall. And I'll put the body wall in here in orange. So this is meant to be the body wall. And what we see here for the left lung is also true for the right. Where there is this continuation of the a continuation of these pleural mesothelial cells, such that it forms the pleural sac. And this is the I don't want to put this here, I got to put something else in there. So this is the intra-pleural space. And taken together, this is the pleural sac. But the pleural sac is composed of two layers. This layer here attached to the body wall, and again, reminding you of the story from the heart, this is the so-called parietal pleura. And then this series of mesothelial cells attached to the lung itself is called the visceral pleura. Now, a couple words here on the pleural sac. The pleural sac is filled with fluid, just as we saw with the pericardial sac. There's this fluid. And just with the pericardial sac, where the fluid acts as a kind of lubricant, again, this fluid acts as a kind of lubricant. Because remember, every day of our lives, we're breathing in and breathing out. And that means this lung has to slip along the body wall structure. And so it slips along the body wall structure back and forth because of the lubrication provided by the fluid inside the pleural sac. There's another important concept, though, here that comes from the pleural sac, and that is that the pressure in the pleural sac is always more negative than atmosphere. We'll eventually see that that's largely because of the elastic nature of the lung and because of the mechanical connection of the visceral and parietal pleura attaching the lung to the wall. This always is being held out, always has a negative pressure. That'll become important a little bit later when we talk about breaking into this sac from the outside world, getting something called a pneumothorax. But let's hold off on that until we get to the idea of elastic recoil. So we have surrounded the lung with these two layers of mesothelium. One attached to the wall of the lung, called the visceral pleura, the other attached to the body wall, called the parietal pleura. Again, the cells have an orientation. The basilar surfaces are attached to the basal lamina. Their apical surfaces face inward to the inside of the sac, and in fact will secrete the fluid that provides the lubrication, allowing the lung to slip back and forth. But that still doesn't tell us very much about how we manage to get air in and out of these tubes and deliver that air down to the alveolar space that we talked about yesterday. So yesterday's discussion was the relationship of the conducting zone with the respiratory zone, and talked about simply blithely moving air in and out of those tubes. What we want to do now is begin thinking about the mechanics of moving the air in and out of the tubes. Now, down here, there's going to be a structure I'm going to put in red. And this structure is muscle. This is the diaphragm. The diaphragm is skeletal muscle. Now, although you don't have to think about your breathing, it's all done automatically. It's automatic, but it's not part of the sympathetic and parasympathetic autonomic nervous system. So there is no innervation of this muscle by either the sympathetic or parasympathetic branches of the autonomic nervous system. The innervation of this skeletal muscle is, as you would expect, from lower motor neurons. And if we come back here and imagine that this is a cross-section of the spinal cord, this would be a cross-section of the spinal cord someplace in the cervical region, probably C2 to C5. We'll see then that there are, and I'll put these in green, there are neuronal cell bodies here located in the ventral horn. It's true on both sides. So these are cell bodies of lower motor neurons. They send processes out into the periphery. These processes enter a structure known as the phrenic nerve. The processes on the right side of the spinal cord find their way out and innervate the so-called right hemidiaphragm. And of course, because we're bilaterally symmetrical here on the left side, these lower motor neurons send a process out into the phrenic nerve, and they are then destined to innervate the left hemidiaphragm. From there on, yes?
Sorry, you have right and left, and then left and right. Is that correct? Or?
I'm sorry, that's a mistake. This should be the left side, this should be the right side, because the lower motor neurons on the left side are going to innervate the left hemidiaphragm. And the motor neurons on the right side are going to innervate the right hemidiaphragm. Okay, thank you. Is that the same question? Right. So what we want to think about now is what happens when we cause action potentials to be elicited in these lower motor neurons. Now, there will be upper motor neurons here. There will be upper motor neurons in the brainstem. They're going to drive these lower motor neurons. And the circuit of the upper motor neurons to the lower motor neurons is running in background on its own. But if you want to, you can override that circuit and hold your breath, at least for a while. And that tells you then that these lower motor neurons are proper voluntary motor neurons innervating proper skeletal voluntary muscle. The action potentials in these lower motor neurons will then release the molecules of acetylcholine. Acetylcholine binds nicotinic acetylcholine receptors. That generates an excitatory postsynaptic potential. This excitatory postsynaptic potential generates an action potential, and you follow through the rest of the story with the release of calcium from internal stores through the DHP receptors and so forth, getting this to contract. Yes?
So I saw my confusion of the left and right. So it looks like the left is getting innervated into the left side, but I thought it was left goes through the right.
Okay, so this is a question about where things cross over. Let's go all the way back there. So remember that it's the upper motor neurons that will cross over someplace in the central nervous system to innervate these. So there will be crossing over of those. But the lower motor neurons innervate muscle on the same side of the body. Just as the lower motor neurons driving your quadriceps muscle, your right quadriceps are on the right side, left quadriceps on the left side. Same story here. And same idea. And I'm sorry I confused the orientation. But so this is the left side. So I'm standing here on right side. And as though my spinal cord here is, then my belly is up toward the ceiling, and you're looking at the left side and right side, which makes this upside down. So let's not think about it. Yes?
Do they first innervate the phrenic nerve and then go to the right and left hemidiaphragms?
So the question here is about the phrenic nerves. We have a right and left phrenic nerve. The phrenic nerves are effectively spinal nerves coming out of the cervical spinal cord. And those phrenic nerves originate from neuronal cell bodies located, I think it's C2 to C5. And those then go out and they will bundle together on the left side and bundle together on the right side. And in that case then, the ones from the left side are going to go to the left hemidiaphragm, and the ones on the right side are going to go to the right hemidiaphragm. Okay? Anything else? So the major idea, irrespective of this squirrely orientation, is that the left side innervates the left hemidiaphragm, the right side innervates the right hemidiaphragm. They obey all the rules that we discussed early on about lower motor neurons innervating skeletal muscle. Then from there on, everything is the same from what we've learned earlier about muscle. When this muscle contracts, what's this muscle going to do? When these sarcomeres shorten, what is this muscle going to do? It's going to shorten. Well, normally that diaphragm is dome-shaped, so it extends up into the thoracic cavity. But then when the sarcomeres shorten, that shortens the whole muscle. So as it shortens and pulls this way, it actually pulls this middle part of the muscle down. Okay? So when it shortens, we'll actually be going from this orientation to more or less a flat orientation. The way I try to show it is we're up like this, we contract it, it comes down. Well, the important idea there is that by using the lower motor neurons to activate this skeletal muscle and getting it to contract, when the diaphragm contracts, it's now going to make the volume of the thoracic cavity larger. We can spell that out. We can say diaphragm contraction increases the volume of the thoracic cavity. So it's a clever trick of this diaphragm and the innervation of the diaphragm to cause the shortening to to shorten the diaphragm and increase the volume of this cavity. So you're okay with that? Because now what we're going to do is going to drill down a little bit and talk about sort of the physical chemistry of this in very simple terms. And this is something called Boyles Law. And Boyles' law simply says that P1 V1 is equal to P2 V2. So that if P1 V1 here is atmospheric pressure and volume, P2 V2 is intrapulmonary pressure and volume. So in the upper panel, that lung space is a closed volume. It's a closed space. This is the atmospheric pressure. The volume of our lung is mighty tiny compared to all of the volume of the ocean of air around us. So this is going to be fixed. We're not going to change that. But what we can affect is this volume here. Because when the diaphragm contracts and shortens, and we said that it increases the volume of the thoracic cavity, so when it contracts, the thoracic cavity gets bigger, that means this volume goes up. So diaphragm contraction increases B2. Well, if this equation is going to remain balanced, P1 V1 has to be equal to P2 V2. If this number gets bigger, what does this number have to do? It has to get smaller. It's the simple algebra of the situation. So now P2 becomes smaller with diaphragm contraction. Well, you recognize here that once we have generated this, P1 is now greater than P2. This is effectively a delta P. It's a pressure differential. Anytime we have seen a pressure differential, according to Ohm's law, delta P is equal to the resistance times the flow. That simply tells us now, depending on how much resistance we have of the air movement along the walls of all of this branching system, we'll get a flow. And that flow now is the movement of air into the lung. So that's the way we get air into the lung. On normal, quiet breathing, we simply contract the diaphragm. The diaphragm is an unusual muscle. It's a muscle that's only found in mammals. None of the other classes of vertebrates have a diaphragm. But as mammals, we have a diaphragm. And we use it now to bring air into the lung. And that's simply done by creating a vacuum and using the pressure differential to move the air in. So now you can imagine that's how you fill your lungs with air. Now it's true, if you start getting down to the weeds of this, once you start exercising hard, you're probably going to have some additional effects of the intercostal muscles, the external and internal intercostal muscles, which also help change the volume of the thoracic cavity. But it's always dependent upon increasing the volume of the thoracic cavity using the passive nature of the delta P to move air into the lung. So now we filled the lung with air, but we also know that we breathe out. So where all of that above and all of this down to here says that breathing in sometimes known as inspiration is active. It requires muscle contraction. But we also find that breathing out expiration is passive. The energy that is used to breathe out, the energy for expiration, is contained in the elastic recoil. Of the lung. So to me, this has always been a little counterintuitive. When you breathe in, it's active. But then when you stop breathing and you stop activating that diaphragm, then the passive recoil is what allows you to breathe out. And if you think about it a little bit, you begin to recognize it has to be this way. Because it's back to the story of sarcomere length. Where if we quickly imagine here, if this were a single skeletal muscle fiber, and there were thin filaments and thick filaments and the whole story that we've discussed so many times, that when the diaphragm contracts, we draw the Z-lines together. We now pull the Z-lines closer together. That imparts energy through the interaction of the actin and myosin to flatten the diaphragm. That energy is then transferred to an increase in volume inside the lung itself, and air comes in. But now we have to figure out a way to reset the sarcomere lengths. We've seen that we can reset the sarcomere lengths in skeletal muscle by using antagonistic muscles. The best example of that, of course, is the biceps and the triceps. You contract the biceps, expand and stretch out the triceps, but now to reset the sarcomere length of the biceps, you use the triceps and reset it. That resetting of sarcomere length in the heart occurred by using the blood filling of the ventricles. Again, we would re-stretch the sarcomere length. We maintain the same problem here, that we have contracted and pulled together the Z-lines, but now we have to reset them. The resetting of these Z-lines in this flattened muscle to the domed muscle, the lengthening of those sarcomeres, comes from the elastic nature of the lung itself. The question then is, where does the energy for this elastic recoil come from? And I'll spell them out, and then we'll walk through them one at a time. The elastic recoil comes from one, elastin in the let's put it so extracellular protein matrix. It also then comes from another source that we're going to see here. Comes from the surface tension in the alveoli. And thirdly, it comes from the negative pressure in the intrapleural space. Remember that the intrapleural space is the space between the visceral and parietal pleura. We said this was always negative. So now we want to see how these elastic recoil characters are managing to allow us to breathe out. The first place I'll talk about this is, you remember yesterday we talked about the alveolar space, and we described how there was this layer of epithelial cells. And we said that this was the air inside the alveolus. We also pointed out that this was a classic form of an epithelium in that there was a basal lamina. And then between the basal lamina of those epithelial cells, there was another basal lamina associated with the pulmonary capillaries. So this is the pulmonary capillary. So we have this basal lamina in here, and this is all extracellular matrix. Among the important components in that extracellular matrix, in the protein structure outside the alveolus, among the important players is the molecule elastin. Remember, elastin was that corkscrew kind of molecule that had the ability, when you pull it, to stretch it out, but it retained the ability, like a spring, to come back to its original position. So, among the important molecules in here will be elastin. If we go back to the upper panel, and I'll put down at the base of one of these alveolar sacs. This is an alveolar sac. And yesterday's discussion was how we moved air in and out. It was the idea that air came in through the conducting region and came down to the alveolar sac, the alveolus, where we had gas exchange. Today's discussion is how we manage to move the air in and out. What's the mechanism? What are the mechanics of moving the air in and out? And what we've discussed so far is generating a slight vacuum by contracting the diaphragm. Air comes in. But now we have to postulate how the air gets back out again. Where does the energy come from to expel the air? Well, some of that energy is stored in the elastin molecules, which are around the alveolus. But there's another source of energy too. I'll put this in purple, but right over the tops of all of these epithelial cells is a very thin layer, no more than, a couple of micrometers or so at most, of water. So one of the things we're going to find here a little later is that because of this water layer, the relative humidity in here is 100%. So a fair number of the molecules in here are water. But what I want to focus on is the liquid water, which is covering over the surface of those epithelial cells. Now you remember when we talked about water early on in the course, we made a big deal of the idea that water molecules had an oxygen, and they had two hydrogens, and that on the hydrogens there was a partial positive charge, and on the oxygens there was a partial negative charge. Then through hydrogen bonding, the partial negative ends of a water molecule could interact with the partial positive charges on neighboring water molecules. Remember, this was hydrogen bonding. That hydrogen bonding will occur among all the water molecules here of the water covering over these epithelial cells. The interaction of these water molecules and their hydrogen bonds generates something known as surface tension. If any of you have ever taken two plates of glass and put a little bit of water on them and then put the two plates of glass together and tried to pull them apart again, you understand the strength of surface tension. This is a remarkably strong force. And under normal circumstances, where the diameter of this air sac is in the neighborhood of about 0.2 millimeters, about 200 micrometers, under those circumstances, the apical surfaces of these epithelial cells are close enough together, they're close enough to one another, that this surface tension actually will bring these cells together and basically stick them together. To illustrate how important this is, at the time of birth, there are a whole bunch of reactions that go on. Because when you're in your mother's womb, you don't have to worry about breathing. All of your oxygen is being supplied from your mother through the placenta. But within a matter of moments after birth, you have to open up this entire airway and bring oxygen and air down into the alveolar space and exchange oxygen and carbon dioxide across this epithelium. Most of the time in utero, there's no need to worry about the level of surface tension in this alveolar space. But what normally happens is, a few days or a couple of weeks before birth, a whole series of events start to occur, and we begin to put into this fluid phosphatidylcholine. You remember phosphatidylcholine has a head group, and it has these fatty acid side chains. It's amphipathic. So among these cells in here, and I should label these blue cells as type 1 pneumocytes. The type 1 pneumocytes are the epithelial cells across which gas exchange occurs. But there's some subset of pneumocytes in here I've shown in orange. These orange cells are said to be type 2 pneumocytes. The function of the type 2 pneumocytes is to secrete these amphipathic molecules, mostly phosphatidylcholine. And why is that important? What it does is these molecules of phosphatidylcholine can have their charged head groups interact with the water molecules. And their tails will then come down here and interact with other water molecules. And effectively, these phosphatidylcholine molecules, because of their amphipathic nature, can insinuate between neighboring water molecules and spread them a little farther apart. As you spread the water molecules farther apart, the distance between the hydrogen bonds gets greater. So the electromagnetic attraction for the hydrogen bonds gets less. So what we're effectively doing, it's a long-winded way of saying the phosphatidylcholine breaks up the surface tension. It reduces the surface tension. So one of the things that happens just before birth is we call on these type 2 pneumocytes to generate a kind of detergent, the phosphatidylcholine, that breaks up the surface tension. And that now allows, it prevents these walls from collapsing one onto the other. You had a question? I know, I misspelled it over here. I was thinking I'd change it for the second one. I knew it looked wrong when I put it up there. It's funny business. Things look different when you're on the board than when you're actually writing on a piece of paper. So we have phosphatidylcholine. The phosphatidylcholine is in here breaking up the surface tension, making it less. So why is that important? Well, it's important because when the air comes into the alveolar space, it expands the alveolar space. It makes it bigger. Now, the first force we talked about to bring that alveolar space back to its original position was the elastin, and that certainly makes a contribution. But also, as we expand this, and we expand that alveolus, we're now increasing the surface area. And as we increase the surface area, we now have a fixed number of phosphatidylcholine molecules interacting with the water molecules. So as we expand it, the effect of the phosphatidylcholine, the surfactant, gets less. And that now allows for a slow increase in the surface tension to help snap that position of this alveolus back into position. So we expand it, and one of the forces working on it is the elastin, but the other is the surface tension, so that when we stop bringing air in, we can now use the energy stored in the elastin and stored in the surface tension to bring this structure back to its original shape. That's part of the passive process. The final one I want to tell you about here. Remember early on, here in the interpleural sac, in the interpleural space here, we said this is always a negative pressure. The easiest way for me to think about this negative pressure is simply that we have mechanically held this elastic structure out against the body wall through the visceral and parietal pleura. So it's basically... Now I want to use an extreme example, it's a clinical example, something that EMTs are really very well aware of. And that is, let's say we have a break from the outside world through the body wall and into this interpleural space. We poke a hole in it. You're in a car accident, it's a knife wound, it's a bullet wound, something breaks this open. So that now, since we said this was a negative pressure originally, what's air going to do? It's going to rush in. What's going to happen now this is atmospheric pressure relative to the intrapleural pressure? They're going to become the same. So now it's no longer negative. It's now the same as the atmospheric pressure. What happens then is, because this is atmospheric pressure, one of the first things that happens is the elasticity here allows this thing to collapse. The other issue is, even when you contract the diaphragm and try to increase the volume here, the path of least resistance for air to come in is through the hole. The path of least resistance for air coming into the lung is no longer to come down through the trachea and all this complicated branched conducting pathway. It's to come through the hole. So two things happen here. One is, because of the elastic nature of the lung, it collapses on itself. And what's more is, we can't reinflate the lung because the pressure here is the same as the pressure inside the lung and on the outside of the world. We can't generate a vacuum. So what EMTs are taught to do is, if they see a chest wound that goes into the thoracic cavity, immediately cover this thing over. Get this thing covered over, and if you can, get a tube in here and basically pull out any air. Because you can pull out the air, then you can reestablish the pressure differential between the inside of the lung and the outside world, allowing you to breathe. But at this point, this so-called collapsed lung occurs because of the problem of the elasticity, which is essential for us to breathe out. Expiration requires that elasticity, but that elasticity now becomes your enemy, because you no longer had this negative pressure inside the pleural sac holding it out. In the last minute here, let me point out one more thing, and then we'll start this tomorrow. And this is the idea of compliance and elastic recoil. And I want to tell this story because we've all seen people with this issue. What happens with people with emphysema? People with emphysema, after years of being exposed to toxins, will break down these elastin molecules. So these start to get broken. The other thing that happens is, when the elastin molecules get broken down and we affect the basal lamina, then these small air sacs, these alveoli, which are only about 2.2 micrometers in diameter, suddenly coalesce and they get bigger. So at that point, with emphysema, we have sacrificed the elastic nature of the elastin, but we've also made these air sacs bigger, so we have now sacrificed the elastic recoil from the surface tension. And so people with emphysema, if you watch them breathe, it's pretty difficult to watch, but they can breathe in just fine. They take a breath. They drop their diaphragm. Air comes in because they don't have much resistance. It's compliant. But they can't breathe out because the elastic recoil's gone. And tomorrow we'll come back to this idea, and I'll give you an example of what we mean by elastic recoil and compliance.